My name is Mike Farzan. I'm a professor at the Scripps Research Institute, and today I'm going to talk about an alternative approach to an HIV vaccine. I'm actually going to be talking about these three molecules. An antibody, I'm sure most of you who are viewing this know what that is, including its antigen binding region and its effector region, the FC region. I'm also going to be talking about CD4-Ig, which combines an antigen FC region with half of the ectodomain of the HIV receptor CD4. And then finally, I'm going to talk about something we developed in our lab called eCD4. And that's nothing but CD4-Ig fused to a small CCR5 mimetic sulfopeptide. Now, we're also very interested in the use of AAV as a means of delivering eCD4 and antibodies to individuals to protect or treat them from infection. This field was initiated by Phil Johnson back in 2009 when he was able to show that if you inoculate intramuscularly rhesus macaques with an AAV vector expressing immunoadhesins, you can protect those macaques, partially protect them, from a rather wimpy SIV MAC239. The field was forwarded and sort of validated by work by David Baltimore, who went on to show that you could, full-length antibodies, AAV could deliver full-length antibodies and protect humanized mice from a complete HIV-1 infection. And in principle, this is a very good idea. AAV can express protective concentrations of antibodies or antibody-like molecules for a long period of time, perhaps upwards of 5 years. AAV itself is considered safe. It doesn't integrate or replicate. So the safety issues associated with AAV come from long-term expression of the transgene. That means you're going to need an off switch, but these are being developed. But there's a catch. The broadly neutralizing antibodies that we in the HIV field consider to be very impressive, very potent, very broad, are not broad or potent enough for this purpose. Broadly neutralizing antibodies neutralize about 60 to 90% of isolates under very ideal in vitro conditions. And what that means is that the IC50 of these antibodies, of 80 or 90% of these antibodies, is less than 50 microgram per mil. 50 microgram per mil is probably higher than what you want to express in your own body. To give you some sense of this, 12 microgram per mil is about 1/1000th of your total IgG. An additional problem with antibodies, in addition to the fact that they're not potent enough, is that it's very, very easy to escape from the antibody. HIV is a pro at antibodies. It knows how to escape. It's seen almost every antibody and has well-worn pathways for escaping from these antibodies. Antibody escape is essentially on the tip of the tongue of every virus. And more problematically, even so, there's no fitness costs associated with escape from antibodies. So this is a problem if you're going to make an AAV vaccine. So if you happen to have the bad luck of being infected with those 1 in 10 viruses that escape from the antibody you're expressing, you will not only be infected, but you'll be an immediate threat to your vaccinated neighbors. It's also more than a problem for therapy. It's nearly impossible for therapy because individuals who are already HIV positive carry a number of resistant viruses in their quasi-species. So most individuals are going to carry a virus that is resistant to most antibodies. There's another problem with broadly neutralizing antibodies that may not be as apparent. They tend to elicit anti-drug antibodies, and in other words, your own immune response to your AAV-delivered antibody, and that anti-drug response can abrogate or will abrogate protection. So AAV-1 is especially immunogenic. It functions as an adjuvant, and it drives the immune response to the antibody that you're delivering. The anti-drug antibody responses correlate with high levels of somatic hypermutations, and the very good HIV antibodies are heavily somatically hypermutated, meaning that they're easily recognized as non-self and frequently eliminated. So what I'm trying to sell you today is that this protein, eCD4, has key advantages over broadly neutralizing antibodies as an AAV-delivered vaccine or therapeutic. It's more potent on average. It's much broader, meaning that it neutralizes everything, including SIV and HIV-2. It's less immunogenic than broadly neutralizing antibodies, and most importantly, it works. AAV-delivered EC4 protects rhesus macaques from very high intravenous doses of HIV-88 and SIV-MAC239, even at titers or concentrations that we can readily achieve in humans with AAV. So some of you viewing this will have seen or read our publication, 2015. So I wanted to highlight those pieces of information that are new in this talk. First, we will show that additional higher doses of the HIV-88 challenge given to our animals, that these animals continue to be protected. We'll also show in vitro data indicating that eCD4 is much harder to escape than a broadly neutralizing antibody or CD4-Ig. I'll nonetheless be able to show you resistant mutants where they emerge that provide the virus partial resistance from eCD4. And then finally, I'll show you protection, very robust protection from SIV-MAC239. I'm not going to have time to mention two other issues that I wanted to flag. One is we've been able to improve the pharmacologic properties of eCD4, and I'm also of interest is our work regulating AAV expression using small molecules. So many of you viewing this probably are aware that HIV enters cells by first association with CD4 and then with a co-receptor, CCR5. And CD4 induces a conformational change in the HIV envelope glycoprotein that allows it to interact with the co-receptor. Now, one thing we discovered a long time ago is that the co-receptor contains at its N-terminus sulfated tyrosines. And these sulfated tyrosines are at the epicenter of the initial interaction between the HIV envelope glycoprotein and the co-receptor. So a little later, we are able to show that antibodies have an inherent propensity also to be tyrosine sulfated in their heavy chain CDR3 regions. So most of you know how an antibody is formed by choosing 1V in the case of the heavy chain 1D, 1J chain, and combining it with a constant region. It turns out that the D region encodes the heavy chain CDR3, and these have a very strong bias toward motifs that are recognized by the tyrosine sulfotransferase that adds the sulfate. So there are now, our original discovery was with a class of antibodies called CD4I antibodies that bind better in the presence of CD4, but there are now a number of antibodies that are shown to be tyrosine sulfated, including of interest apex antibodies to the V1, V2 region. And I've listed those here. Of the tyrosine sulfated CD4I antibodies, two are especially important. The first is 412D, and this is important because Peter Kwong solved the structure of this antibody, which I'm showing you here in its light chain, its heavy chain, bound to HIV GP120 and to CD4. And that was very important for us because it allowed us to map where the sulfotyrosines bound to the HIV envelope glycoprotein. So we now know where these highly conserved sulfotyrosine binding pockets are on the envelope glycoprotein. A related antibody was also important. Its name is E51. And what's important about E51 is if we take the CDR3 region of E51, turn it into a peptide and fuse it to an FC region, we're able to use that construct to precipitate GP120. So if you look on this gel, you can see GP120 over here with this just run as a free protein. And then E51 precipitates GP120, but not any of these other CD4I sulfated or unsulfated antibodies. I should say the heavy chain CDR3 regions as peptides. So now the reason this works is that the CDR3 region of E51 looks very, very much like the amino terminus of CCR5, and in fact it has the same pattern of sulfotyrosines as you can see down here. However, it's lost some of the insoluble or complicated regions of CCR5, like its O-glycosylated region, and hydrophobic amino acids that point down into the body of CCR5. So it's more soluble, behaves better, but mimics the same pattern of sulfotyrosines present in CCR5. As you would expect, this peptide, which we refer to as CCR5-MIM1, precipitates GP120 more efficiently in the presence of CD4. So you can compare this band to this band, this band to this band, this band to this band, et cetera. So that suggested to us that we should fuse CD4-Ig, a compound used in the clinic, safe but not effective, with our CCR5 mimetic peptide. And we did so in 3 ways. And to make a long story short, the best fusion was where we put the peptide at the C-terminus of CD4-Ig. And when we did so, we significantly increased the potency of CD4-Ig. So for example, here, I'm showing you in red neutralization by CD4-Ig, and then neutralization by eCD4-Ig. And you can see what we've seen now continually, a 1.5 to 2-fold difference, improvement in the ability of eCD4-Ig to neutralize various HIV and, importantly, SIV isolates. You'll notice in this panel SIV MAC316. This was our first indication of that eCD4-Ig was going to be unusually broad. So we then turned to Mike Seaman and asked him to analyze 2 sets of hard-to-neutralize viruses and compare CD4-Ig to eCD4-Ig and several broadly neutralizing antibodies. These panels include a diverse panel of Tier 2 and Tier 3 isolates, and it also included a comprehensive set of isolates that are resistant to what were then the 2 best neutralizing antibodies, NIH-4546 and 3BN-C117. And this figure summarizes a series of experiments, neutralization studies, in which we're comparing, in red, again, CD4-Ig to eCD4-Ig, and now 3 different variants of eCD4-Ig, all varying just with a single amino acid mutation. And for the purposes of this talk, these differences don't matter. You can see they behave approximately the same. And then to the CD4 binding site neutralizing antibodies, VRC01, NIH-4546, or 3BNC. The first thing you can see is that 1 to 2-fold drop improvement in neutralization efficiency relative to CD4 for all our eCD4-Ig variants, and about 1 to a half a log drop improvement relative to the best CD4 binding site antibodies. More importantly, however, is that there is simply no isolate that we've tested or that we're aware of that's resistant to eCD4-Ig, whereas there are a number of resistance viruses that are resistant to CD4-Ig and, of course, to neutralizing antibodies. The summary of this, so the variant of eCD4 that we like the best, we call eCD4-Ig MIM2. It simply incorporates an extra tyrosine in the sulfopeptide. It has a geometric mean IC50 of 0.05 microgram per mil. This number was based on hard-to-neutralize isolates. Importantly, all IC50s were less than 1 microgram per mil, and all IC80s were less than 5.2 microgram per mil. And these, again, are concentrations that one can easily achieve with AAV expression, intramuscular AAV expression. Again, 100% of the isolates were neutralized, including several SIV and HIV-2 isolates. So why is this thing so potent? Well, there's 3 different reasons. First of all, eCD4-Ig differs from CD4-Ig in that it does not, like CD4-Ig, enhance infection. So CD4-Ig does this by promoting direct association of the envelope glycoprotein with the co-receptor, bypassing the need for cellular CD4. eCD4-Ig sulfopeptide blocks the interaction between the envelope glycoprotein and the co-receptor, and it abrogates this tendency of CD4-Ig to promote infection under some conditions. However, like CD4-Ig, eCD4-Ig irreversibly inactivates the envelope glycoprotein. And in fact, eCD4-Ig may do this better because it has higher occupancy, and it may mimic some of the tendency of the properties of CCR5 to promote further conformational changes in the envelope glycoprotein. But perhaps the most important reason for eCD4's potency is its avidity. Most antibodies bind the envelope glycoprotein with one of its 2 arms. eCD4-Ig has 4 arms, and at least 3 of these are engaged when it recognized the envelope glycoprotein. That's both of its sulfopeptides and at least one of its CD4 components. Why is it so broad? Why is eCD4 so broad? Well, this is probably more intuitive. Antibody epitopes are always bigger than the receptor and co-receptor binding site. eCD4-Ig binds only functional and necessarily conserved regions of OMV. Also, every virus has escaped every type of antibody in its recent history, and it remembers that in a way in the form of well-worn, easily accessible escape pathways. The virus, in contrast, has never seen eCD4-Ig, and there's not an easy way for the virus to escape. And we show that here with in vitro studies in which we passaged for 200 days, over 80 successive passages, several swarms of virus in the presence of the antibody NIH-4546, CD4-Ig, or eCD4-Ig, or without inhibitor at all. And we did so in independent wells. That's why there's, for example, 5 lines here. So that's representing 5 wells. We then asked the question, how do these swarms, after they've been passaged in this way, react to each of these 3 reagents? So you can see that in the absence of inhibitor, when viruses passage in the absence of inhibitor, eCD4-Ig, as with the original virus, is more potent than both CD4-Ig in red and the antibody in green. When we passage in presence of NIH-4546, a broadly neutralizing CD4 binding site antibody, you can see that we now get ready escape from that antibody. However, the virus remains sensitive to both CD4-Ig and eCD4-Ig. Now, if we passage in the presence of CD4-Ig, we easily get escape from CD4-Ig. However, we don't get escape from eCD4-Ig or expectedly the antibody 4546. Now, finally, what happens is when we passage in the presence of eCD4-Ig is that we get escape from CD4-Ig, and we shift the curve but do not get escape from eCD4-Ig, and the virus actually becomes a little bit more sensitive to NIH-4546. So we use this data to conclude that, at least in vitro, eCD4-Ig is much harder, viruses have a much greater difficulty escaping from eCD4-Ig than they do from CD4-Ig or from a very good neutralizing antibody. We took the virus that was partially resistant to eCD4-Ig from the previous study, sequenced it, and I'm showing you in magenta where the resistance mutations have emerged. As one might expect, show up in the CD4 binding site, presumably lowering the affinity for CD4, and then also in this critical sulfotyrosine binding pocket, as you can see here, indicating that the virus is attempting to resist eCD4-Ig by lowering its affinity for that particular sulfotyrosine and again for CD4. We then did in vivo studies in an effort to show that eCD4 could protect animals from a SHIV challenge. What we did was we generated AAV vectors that expressed rhesus form of eCD4-Ig and a rhesus form of TPST2. TPST2 is an enzyme that sulfates the eCD4-Ig and CCR5, and in this case it is necessary because when you overexpress a protein, you overwhelm the endogenous sulfotransferase activity of the cell. So we needed to provide TPST2 at about 20% of the level we were providing a vector expressing rhesus eCD4-Ig. So we inoculated 4 rhesus macaques at a fairly standard level of AAV-1, and again at a 4-to-1 ratio between eCD4-Ig and TPST2. The animals were challenged repeatedly with escalating doses of SHIV-88 until they were infected, or at least until all the controls were infected. And then we analyzed the serum for protein titers, viral loads, and immune responses to eCD4-Ig. What we observed after some time is that 2 of our animals expressed stable concentrations of eCD4-Ig, stabilizing at about 70 microgram per mil, and another 2 animals expressed approximately 15 micrograms per mil of eCD4-Ig. The second set of animals are important because that, again, is approximately what you might achieve in humans and might want to have expressed in humans. So any protection that you would see in animals at these concentrations may be relevant. We also analyzed the anti-drug antibody responses to eCD4-Ig and compared it to animals that were seeing rhesuscized forms of these different broadly neutralizing antibodies. And what you can see is that that ECD4 is much less immunogenic than any of these antibodies, despite the fact that they carried rhesus constant regions. However, if we scaled this signal up, we could see that 2 of our animals did indeed generate anti-drug antibodies. These anti-drug antibodies did not correlate with their titers and they did not appear to interfere with the expression of the protein, but they are indeed there. We then challenged our animals with escalating doses of HIV-1. Red, again, is our control, and blue is our rhesus eCD4-Ig inoculated animals. When we challenged with 200 picogram, P27, of HIV-1, 2 of our control animals were infected. We challenged again with 400, and our third control animal was infected. Our last control animal was kind of recalcitrant, so it was not infected with another 400 picogram challenge, but was infected with an 800 picogram challenge. At this point, you can see that none of our rhesus eCD4 animals were infected. Now, that was published. What's new is we continue to double and then quadruple our challenge doses, and again, our rhesus eCD4-Ig inoculated animals remained seronegative. So we went ahead and performed a very similar study using the SIV-MAC239, only we had 8 control animals instead of 4. We wanted to do so because we thought it would serve 2 purposes. First, it would establish clearly the breadth of the in vivo protection that eCD4-Ig is providing. And secondly, SIV-MAC239 is considered a gold standard virus. It's well adapted to its rhesus host, and we thereby circumvent some of the difficulties associated with the use of shivs. Same experiment again, only we got a little bit lower titers this time, hovering around 25 micrograms per mil and about 5 micrograms per mil. In this case, 4 of our 4 animals saw an anti-drug eCD4-Ig response, which went away nonetheless quicker than in our previous experiment, except in one case. Again, though, these anti-drug antibodies were relatively modest compared to that which one sees with neutralizing antibodies, and they did not interfere with the expression, appear to interfere with the expression of the transgene. We analyzed where those anti-drug antibodies were occurring, and as we had seen in our previous animals, there were no anti-drug antibodies to the rhesus CD4 ectodomain, so we're not recognizing rhesus CD4. Some of our animals in this group, as well as some of our animals in our previous group, had anti-drug antibodies to the junction between the FC region and CD4. However, in this group, in contrast to the previous group, our animals had clear antibody responses to the peptide, or the peptide Ig. We don't know the reason for the difference between this group and our previous group. It's exactly the same protein. The work was done at 2 different primate centers, but there was a very clear difference. Nonetheless, we went ahead and challenged those animals with, at this point, 20 and 40 picograms of SIV-MAC239. You can see all 8 of our challenge monkeys were infected, and none of our rhesus ECD4-Ig inoculated animals were infected, suggesting that we have very robust protection against SIV-MAC239. So our take-home is that ECD4-Ig is a very good inhibitor, on average more potent than the best neutralizing antibodies, and much, much broader than any antibody. It is more difficult to escape than antibodies in vivo. We hypothesize, but don't know yet, that escape virus will be less fit and less transmissible. It is also much less immunogenic in macaques than heavily hypermutated broadly neutralizing antibodies. When expressed with AAV, it provides robust and consistent protection from two highly divergent strains, HIV-88, a Tier 2 virus, and SIV-MAC239, a Tier 3 SIV virus. If it continues to live under the immune radar, it could protect most humans from most potential transmission events. Its use as a therapeutic, replacing or supplementing some components of ART, remains to be demonstrated, and we should have data on that point this year. However, the safety of this approach as a vaccine or therapy has not been established, and an off switch would be necessary for its broad use. Nonetheless, this general approach is closer to realization than any conventional vaccine effort. And then I would just want to acknowledge the individuals who contributed, especially Matthew Gardner, who led our entire eCD4-Ig effort. Christoph Ellinger did the escape studies that allow us to determine where the virus attempted to escape from eCD4. And then I haven't had a chance to talk about our off and on switch efforts, but Guozai Zhang and Huihui Mo are both important for those. And thank you.